So today I want to talk a little bit more, give you more insight in what sort of learning is going on in the brain, so how the brain controls the bladder. There are significant knowledge still in how the brain controls the bladder, constitutes that information. Lower urinary tract symptoms affect many people. And that's can result from various diseases, including neuronal. So it's really important that we improve the understanding of neural control and circuits to identify novel therapeutic targets and to improve Okay, so the storage reflex is thought to be mainly intraspinal. When the bladder stretch signal during bladder filling, the bladder stretch signal comes in to the sacrospinal cord. And there are sensory interneurons that then send that signal to the thoracic spinal cord where the sympathetic bladder motor neurons are. And the sympathetic outflow allows for contracting the bladder outlet and relaxing the detrusor, so to accommodate for filling, for storage. Then the voiding reflex requires supraspinal brain nuclei. The bladder effect signal, again via a sacrospinal cord, is sent out very active to the brain. That in turn then activates a center in the brainstem called the multi-micturation center. And activation of this micturation center sends a signal down to the sacrospinal cord again to induce voiding, so to contract the bladder and to relax the external urethral sphincter. And this multi-micturation center is really important. Already 100 years ago, Dr. Barrington made lesions in the brainstems of cats. And he found that the cats couldn't be anymore, they retained. But still, 100 years later, a lot is unknown about the region, the cell types, the pathways, how the brain controls the lower urinary tract. So this is an image of the mouse brain atlas. It's a coronal section, and you can see the multi-micturation center, that is also known as Barrington's nucleus now. On both sides of the brain, underneath the cerebellum, so in the very far back of the brain. And we use mouse lines, mouse models, because that gives us a handle, it gives access to different neuronal populations to study them. So here you see a brain section of a V-GLUT reporter mouse. And all the glutamatergic V-GLUT2 expressing neurons there are green. So you can see in Barrington's nucleus, most of the neurons there are green. As opposed to in a different mouse line, the VGAT reporter mouse line, there are almost no GABAergic green neurons in Barrington's nucleus. So most of the neurons in Barrington's nucleus are excited. And from Barrington's nucleus, there are these neurons that project down to the sacrospinal cord to innervate the bladder and the sphincter motor neurons. So we traced the projections of the axons from the brain to the spinal cord. And here you see at different levels where they terminate. So mostly in two regions, in the intermediolateral subcolon, which is where the parasympathetic bladder motor neurons are located on the sides, and in the dorsal great commissure in the middle. And in the dorsal great commissure there are interneurons that connect to the sphincter motor neurons in Owner's nucleus in the ventral horn. There are actually no direct projections from Barrington's nucleus to Owner's nucleus for sphincter control. And we also tested the other way around. So we injected a retrograde tracer into the spinal cord, and we found out, we saw what was labeled in the brain, so what is projecting to the spinal cord. And we did that in a CRH green mouse line. So here the green neurons in Barrington's nucleus, they are CRH expressing. And then the red neurons, they are projecting to the spinal cord, and if they're both, then they are CRH neurons that project to the spinal cord. So quite a lot of the Barrington's neurons are CRH expressing, and they are spinally projecting to control the motor neurons. We went on and looked at how the neurons in Barrington's nucleus, how they activate during filling and voiding. So we used the calcium center that measures the changes in calcium levels, and we used it as a proxy for neuronal activity. while looking at bladder pressure at the same time. So we started with the glutamatergic neurons, because most of the neurons in there, because nucleus are inhibitory. The orange trace is an average of multiple void events, and then the dashed line is the moment the start of voiding. And we looked at the 60 seconds, a minute before the void event and 60 seconds after, and how the neurons activate the whole glutamatergic population, how that activates during this time. And you can see that these neurons start activating already before the pressure is going on. So we think that they drive the pressure to the bladder to contract, the pressure to go up. And then if you look in the CRH subpopulation, subgroup of all the glutamatergic neurons, they show a slightly different pattern. So they activate later. Their activity goes together with the pressure going up. So that supports a role for these neurons in promoting, keeping the detrusor contracted and promoting bladder contractions. So as you can see, the glutamatergic activity in the glutamatergic neurons is longer, the curve is wider than the subpopulation. So we wondered if there are other populations in Barrington's nucleus that maybe have different functions for blood control. So we went on to perform single nucleus RNA sequencing. We took a small punch, a small dissection of around Barrington's nucleus, and then we dissociated the tissue and got a single nuclear suspension that we ran on a special device. So we ended up sequencing the barcoded messenger RNAs within those cells to get to this clustering analysis, as Jim was already nicely introducing this. So cells that are more similar, transcriptomic-wise, they cluster closer together, and cells that are more different, they cluster farther apart. So astrocytes are different from neurons, and the neurons cluster together. And then we took out from all these cells, we took out only the neurons, the glutamatergic neurons, and we clustered those again to see if there were some, or the clusters were. And we found that there are 22 different neuronal populations. But that is within the bunch. That is within the whole 1 millimeter circle. So that is a little bit more than only Barrington's nucleus. And this dot plot shows marker genes that are expressed within cells of the specific clusters. So if a gene is highly expressed in only one cluster, then that is a good marker to target that population to study it further. Okay, so we know what all the marker genes are in all of these clusters. But a disadvantage of single nucleus RNA sequencing is that you lose the spatial information. So after the dissociation of the tissue, you have no idea where the cells were before. So we looked online in the Allen Brain Atlas in situ hybridization experiments, or we did RNA scope ourselves to find out where the cells are that express these markers. And this is an example of CRH, and you see that cells expressing CRH, neurons expressing CRH, they're very nicely located in specific to Barrington's nucleus. And then for all the populations that we found that are in or very nearby Barrington's nucleus, we checked if there are transgenic mouse lines available so we can target them. And if they're not available, then we need them. So for example, for this study, we generated 2 new mouse lines. So everything is possible. We then, for the neuronal populations that reside in Barrington's, we traced the axonal projections to the spinal cord to see if they could innervate the motor neurons there. And here you see a horizontal section of the spinal cord. So in the CRH mouse, we injected a tracer, and you see the CRH neurons in Barrington's nucleus being labeled. And then we followed the red axons all the way to the lumbosigmoid spinal cord. In the IML is where the cholinergic bladder sympathetic, parasympathetic motor neurons are to contract the bladder, and in the dorsal great commissure is where the interneurons are that connect to the sphincter. So these neurons specifically project to that L6S1 level to innervate those neurons. For a different neuronal population, bank, chlorothalonil, the neurons are also labeled in Barrington's. You see them being labeled. And these neurons also project their axons to the same level, both to the IML and to the dorsal curvature. And this is another neuronal population that we found, and there are also cells in Barrington's nucleus that express this marker, TAC1. The projection pattern is a little bit different. So these neurons project at many different levels along the spinal cord, and also more caudally, more at S3, S2, S3, S4, sacral levels. But they also innervate a little bit the CNS or IML. It's a different pattern, different population. So we identified 4 of the populations that reside in Barrington's nucleus and that project axons to the sacral spinal cord. We also tested their functional contribution to that function. And for this, we used a technique called Tract, chemogenetic stimulation. So Tract stands for designer receptors exclusively activated by designer neurons. These muscarinic receptors, they are modified, so they don't respond to the endogenous ligand anymore, but instead they respond to a drug, CNO. You give it systemically, and it can activate or inhibit intracellular pathways, and thereby activating neurons or inhibiting neurons. And then we looked at the behavior of these mice on our voiceful assay. We use a thermal camera that is overhanging behavior cages, and we record the behavior, so afterwards we can look back at the video and see what the mice did. So here we injected bilaterally in 2 sites this excitatory, stimulatory threads in the Barrington's nucleus of a bank mouse to activate specifically the bank neurons. And you see a screenshot. This is at the end of a 2-hour run. The mouse peed 1 time in the corner. This is totally normal looking. But then when we activated the cells by giving CNO, the mice suddenly started spotting, leaking all over the cage, irregular places. So even though stimulating these bank neurons does not really change the number of voids, it does significantly change, increase the number of leaks that these mice are doing. We also recorded the neuronal activity in these specific, in this subpopulation of neurons. And the dashed line is the onset of voiding. So these neurons, they activate at the start of the voiding. And they're active only for the duration of the voiding. So during voiding, the external urethral sphincter must be relaxed. So we thought that maybe these neurons play a role in controlling the external urethral sphincter. So we tested that, and we implanted electrodes in the sphincter, and we stimulated these neurons using optogenetics this time. Because optogenetics is a lot more temporary sensitive, you'll see. So we expressed a channel rhodopsin, a light-sensitive ion channel, into the brain of these mice. And then we implanted an optical fiber to deliver the light. And we recorded the sphincter activity on EMG and the bladder pressure on the CMG. And here's where we give the light for 10 seconds. And we look at the activity of the bladder and the sphincter, and you see that the sphincter activity is significantly reduced during the light stimulation. So light activating these bank neurons reduces the activity of the sphincter compared to before and after the stimulation, but there is no change to bladder pressure when we activate these neurons. So bank neurons and Barrington's nucleus directly control the external urethral sphincter. The bank and the CRH population, they're separate. They do not overlap. Only 1% of all the Barrington's neurons express both bank and CRH. And when we light stimulated the CRH neurons, we saw an increase in bladder pressure. And also, Lisa Stauer's group at Scripps, in their 2018 paper, the upper trace is a bladder pressure trace, the lower trace is a sphincter activity trace. They also saw an increase in bladder pressure, but no change in sphincter activity when they stimulated the CRH neurons. So I showed that there are excitatory neurons in Barrington's nucleus that activate around the time of voiding. And the Barrington's nucleus contains different neuronal populations. So distinct subpopulations play specific roles while innervating the spinal motor neurons. So the CRH neurons play a role in contracting the bladder, and the PANC neurons play a role in relaxing the external urethral sphincter. So we also investigated how Barrington's nucleus itself is getting activated. So we mapped out all the inputs in the brain that innervate Barrington's nucleus to see what can control its activity. This is a very large list. So we used several retrograde tracers to find out which brain sites could control Barrington's activity. And one of these sites is the periaqueductal brain. And in there are GABAergic neurons, inhibitory neurons, that send direct projections from the PAG to Barrington's nucleus. As you can see here, in a vegan mouse, so a GABAergic mouse, a tracer injected in specifically the GABAergic neurons, and you see the axons terminate in Barrington's nucleus. So the GABAergic neurons in PAG, they connect with Barrington's nucleus. So we hypothesized that these neurons would be able to control the activity, could inhibit the activity of Barrington's neurons. So we live activated them. In this case, actually, we injected the generalopsin, the activator ion channel, in the PAG, and the optical fiber was implanted on the axon terminals. So the axons that were sent from the PAG neurons to Barrington's nucleus, only the Barrington's projecting axons were activated or stimulated. And then we infused saline, we ran these mice on CMG. Every peak is a void contraction, and you see that a cycle on average takes like 10 minutes. When we were expecting the next void to happen, so after 9 minutes, we started the stimulation lasting 10 minutes. And during the whole time that the light was on, the mice didn't pee and couldn't, there was no void contraction and they didn't void. And then when the stimulation was stopped, there was almost immediately there was a void contraction again and the mice peed. And then the regular interval resumed. So clearly the duration of the cycle is extended. Activation of these inhibitory neurons, inhibitory in the Barrington's nucleus, can postpone voiding. If we compare the cycle in which we stimulate it, it's extended. So stimulation of the GABAergic neurons in the PAG that connect to Barrington's nucleus can prevent activation of Barrington's nucleus, and thereby help maintain continence. So we identified all of the neuronal populations in Barrington's nucleus, also around Barrington's nucleus. And so Barrington's nucleus is important for initiating icteration. And we identified which of those project their axons to the lumbosacral spinal cord. And we tested the functional contributions for the different populations for that function. And since activity in Barrington's nucleus is essential for coordinated voiding, so for simultaneous contracting the bladder and relaxing the sphincter, we showed that the inhibitory neurons that can control them, that they can control the activity. So we are developing a blueprint for the neural control of blood function by revealing the molecular identity of the neuron populations and all the circuits that are connecting them. And similar as for the Barrington's nucleus, we are also performing single nucleus RNA sequencing for the spinal cord, for the mouse spinal cord, and for the human spinal cord. So to reveal the identity, the genetic identity of all the neuronal populations in the spinal cord as well, that may be downstream of Barrington's neurons, to find their exact location and to see if they're involved in blood function, and then to see which of those mouse neuronal populations are evolutionarily conserved in the human, and which could be important for human blood function. So the aim is our goal is to dissect the neural circuits and to identify targets that hold high potential for better, good therapeutics. And with this, I would like to thank the people in my lab, Natalie, Mihogo, Andrea, and Richard. collaborators, Marcelo and other mentors at BIDMC. Important research core facilities that have helped us along the way, and the NIH for continued funding. And Chris for inviting me here.